Good morning on this rather cold start of December. I'm Bonnie Raindrop, and I'm the project director for Pesticides and the Chesapeake Bay Watershed Project, now in our 17th year. This project is a program of our organization, the Maryland Pesticide Education Network, MPEN for short. We want to welcome you to this first of two conference webinar sessions. We're delighted that this year we have over 200 stakeholders registered for the conference. Many of you are longtime friends who have been part of this community and have attended these conferences year after year. Welcome back. Some of you are members of the project's working groups and help advance a variety of projects with us. We also have some first-timers, and we're especially glad to have you joining this community of professionals. All of us are concerned in some way with the impacts of pesticides on the Bay watershed. You are all experts each in your own right. And the Watershed Project is about stakeholders coming together to learn and collaborate, even virtually, to advance our understanding of the role pesticides play in the watershed and how we can work together to more effectively address pesticide impacts. Thank you for taking time to be with us today. Next. Before we move on, please open your chat window with the icon at the bottom of your Zoom screen. Use the little arrow to select Chat to Everyone. And please introduce yourself and share something about you. I think we all miss the networking that we used to do at this live conference, but we'll do the best we can with chat. You can also change the toggle on the chat icon so that you can chat with an individual attendee or chat to the presenters only. So please chime in now and share your thoughts today as we hear from our presenters. I'll be sending a chat transcript after the last session, and you'll also get the roster of all the attendees with contact information to help you with further networking. Use this chat like a networking opportunity when you start to post. And maybe the post about your special areas of interest may help you connect with a new colleague. We'll also use the Q&A module to pose questions to our presenters. So click on that Q&A icon at the bottom of your screen and type questions in as we go. Remember, if the question's for a presenter or a panelist, you must use the Q&A tool. And putting these questions in the chat won't get them answered by the presenters. If we don't get to your questions live, our presenters are going to stay on after the Q&A to respond in the Q&A tool, and we'll send that transcript along with the chat transcript next week. At around 11:30, we're going to pause for 5 minutes so you can take a short break after our keynote. Next. Now, I want to turn things over to our conference moderator, Dr. Greg Allen. We're really proud that Greg's been our moderator since its beginning 17 years ago. And Greg also has led the Research and Data Gaps Working Group for just as long. And now he shares that role with Dr. Vicki Blazer. Greg is an environmental scientist at the U.S. Environmental Protection Agency, Chesapeake Bay Program Office, and is approaching a 30-year anniversary at EPA. He's chair of the Chesapeake Bay Program's Toxic Contaminants Work Group and coordinator of the Partnership Leadership and Management Team. For nearly a decade before joining EPA, Greg was in the field and at the analytical chemistry bench, analyzing the occurrence of organic contaminants. Greg counts among his most meaningful recognitions a White House Closing the Circle Award, an EPA Gold Medal, and a handwritten thank you note from the Puget Sound Partnership for helping with their PCB reduction strategy. Welcome, Greg.
Thank you so much, Bonnie. Thank you, and good morning, everyone. Welcome again to our 17th annual Pesticides in Chesapeake Waters Conference. I know I am so excited about this year's theme. That theme is healthy soils and healthy soil microbiomes. And we have some phenomenal speakers to talk to us about that. I know this morning we were commenting on, wow, could this really be our 17th? And doesn't time just fly by? Yes, but here we are. And I think there are a few things that have made this project extra special. One of them is that we have these work groups that you'll be hearing from both today and at our second session in a couple weeks. You'll be hearing from our work groups. And this project has really been special because these work groups come together and produce products. And I personally think that's one of the things that makes it special. We have generated a range of products out of our work groups that are hopefully really helping inform the community in the watershed about pesticides. So really have always admired that. We're going to keep that going. And I think the other thing that has helped to sustain this project over time is the important values that we all bring to the project, and in particular, that we bring to this conference. And so we're seeing those on the screen here, and I'm going to pause for a moment so that you can read these values that help to sustain our project. All right, so these are our shared values that make sure that we have really constructive conversations in the conference and in our work group proceedings. And I'm sure it'll be the same this year, that we'll use these values to be respectful of all perspectives. So, okay, with that, we're going to shift over here to a question that we have for you. So we've built in lots of chances for you to be engaged and participate today. And here's one of them. We got a chat question for you. I'm hoping you'll use the chat button that you should see on your screen. I think most all of us are experts in this stuff at this point. So hope you'll find that chat button and open it up and share with us your favorite place or favorite activity that you enjoy in the Chesapeake Bay watershed. Or if you're joining us from outside the Chesapeake watershed, something that's a favorite place or activity where you live. And I'm going to pause so that folks can enter some thoughts into chat, and you'll have a chance to read some of them. Wow, thank you, everybody. This is amazing. These are wonderful activities: hiking and biking, kayaking, canoeing, sailing, incredible places, parks that we have, conservation areas, exploring all the nooks and crannies in the watershed on bike and by kayak or canoe. Yeah, even some folks treasuring some of our most troubled areas like the Anacostia, which is recovering. Oh, and our spectacular Bay Bridge. Yes. All right, great. Well, you can continue to add your thoughts here, favorite places and favorite activities. See some Annapolis love in there. Thank you. Mm, wow, these are some places I haven't seen. Hopefully we're all getting a little inspired here about some things to do and places to go visit. There's just so many beautiful places, and it's an amazing resource, this Chesapeake Bay watershed. And we all come together to try to understand some of the risks that are presented to it and see what we can do to work together to manage those risks. All right. Really excellent places and activities. Thank you all for doing that. And you can continue to add and be inspired by that list in chat. Okay, well, we'll keep moving here. Next, I'm going to give you a little overview of today's agenda. So when we think about climate solutions, we might not immediately think about the soil microbiome and the role that it plays, but it really is an important element. And yet last year we had a presentation from a Maryland farmer, that was Mason's farm, and it is the largest organic grain producer in the state. And we heard about that farm's implementation of organic regenerative farming practices. It really generated a lot of interest because it's a positive example that shows us that it's feasible to have a profitable farm that uses regenerative farming practices. And so this year we wanted to build on that sort of solution thinking with our sessions around the soil microbiome. So we have a couple of experts that have very deep experience with both the soil microbiome and the human microbiome, and the potential impacts that pesticides can have on those microbial communities. So we're going to kick things off today with a report from our project's Research and Data Gaps Working Group. And then we'll go over to our keynote speakers. And just so lucky today to have the time of Elaine Ingham and Adam Cobb talking to us about soil health and that it is critical to our future. And then we'll have Kendra Klein, and she'll talk about genetically engineered soil microbes, risks, and concerns. So really great program. And we're going to kick it off here with the Research and Data Gaps Group.
Well, we are really delighted to have 3 longtime working group members with us. In addition to Greg, we've got Dr. Vicki Blazer, who's a research fishery biologist with the U.S. Geological Survey at Eastern Ecological Science Center, Leetown Laboratory. She has an undergraduate degree in marine biology from Southampton College and a PhD in fisheries, aquaculture, and pathology from University of Rhode Island. Vicki's research is currently focused on risk factors such as pesticides, endocrine disruptors, and other environmental contaminants and their influence on both infectious and non-infectious fish disease and fish populations. Her lab focuses on histopathological, immunological, and genomic bioindicators of exposure. She works closely with both federal and state resource agencies throughout the U.S. to address specific issues in fish health, and also has international collaborations. She's been an adjunct professor at West Virginia University, Penn State, and University of Maryland, where she serves on graduate committees. She's authored or co-authored over 200 peer-reviewed journal articles and has served on numerous professional society committees or elected positions. Dr. Ian Hartwell has more than 30 years of training and experience in freshwater, estuarine, and marine biological systems. He's most recently was the chief scientist for the NOAA Status and Trends Bioassessment Projects for the National Oceanic and Atmospheric Administration. In his role, he managed long-term monitoring and assessment programs in estuarine and coastal systems at various locations around the U.S. As lead scientist, he worked on industrial new chemicals for potential ecological effects for EPA and Superfund assessments. He was a postdoc at University of Maryland Horn Point Lab, spent 10 years as director of the Maryland Department of Natural Resources, Chesapeake Bay Contaminants Assessment Program, and 20 years as NOAA Status and Trends Bioeffects Program Chief Scientist. Ian has a BS in Fisheries Management from University of Michigan, an MS in Biological Oceanography from North Carolina State, and a PhD in Environmental Toxicology from Virginia Polytechnic Institute. We're very honored to have Greg, Vicki, and Ian representing this project's Research and Data Gaps Working Group. Vicki, I think you're going to start things off.
I am. Sorry. So what I'm going to do is just go over what we have been busy with this past year, and that's finishing up this report on communicating recent pesticide research findings to inform stakeholders. The purpose of this report is really to compile and review relevant scientific literature. We focused on the period from 2010 through 2021 because basically it's a follow-up for the report that was published in 2009. The previous report, which was shared with many project stakeholders and the government's cabinet, and we hope that the same will continue for this one. So in this, we summarize the research for use. And again, we hope that it will be used not only by this conference, but state agencies, legislatures, and concerned citizens. Next slide, please. So what this provides is an overview of findings on key research studies, published literature, and also identifies data gaps. We provide 6 recommendations for potential actions to the Bay cabinets and other stakeholders. So we've summarized, and these are just brief paragraphs of summaries of the studies of over 25 studies that we felt specifically related to the Chesapeake Bay watershed. Next slide. What we hope is that these research findings inform strategies to reduce the impacts of pesticides on the Bay watershed and its life forms. As I mentioned, it's sort of summarizing research for the past 11 years. What we hope to do is not wait 10 or 11 years to get reports out, but to update this effort annually and keep current with the latest research findings that can improve both strategies and policies. Next slide, please. So just to give you an idea of some of these papers, I do want to emphasize that when I say we summarize 25 studies, as you might guess, there are hundreds of papers out there on pesticides and effects on various organisms, pesticides in soils, all those sorts of things. What we focused on was ones that we felt had the most relevance to Chesapeake Bay, even though they may not be or have been conducted within the Chesapeake Bay watershed. So each of us, Greg, Ian, and I picked a paper just to give you an idea of why we felt these were important and what's in the report. So my choice was a paper by Mahler et al. It was published in 2021 in Environmental Science and Technology, where they evaluated 108 parent compounds and 116 pesticide transformation products in small streams from 5 regions of the US. This is an example of a study that was not specifically done in the Chesapeake Bay watershed, but is very relevant. The findings are very relevant to what we are seeing. Transformation products or degradates of pesticides are formed by many processes. And so they'll vary depending on water quality, climatic factors, all sorts of things that affect those processes. What this paper found and others have found is that concentrations of these products often exceed the parent compounds. One of the studies that they actually cite in their paper shows that 70% of these are as or less toxic than the parent compound, but 30% are actually more toxic than the parent compound. But these compounds are not routinely measured. And so it is a data gap, I think, that it identifies. The graphs on the right-hand side of the slide are actually not from this paper, but are from some of our data collected within the Chesapeake Bay watershed. And it's just to illustrate that we are seeing the same things and that it varies from site to site. And I chose atrazine and one of the atrazine degradates just to illustrate. Within atrazine, we commonly measure 3 to 4 different degradates in the water. But what you can see is that our Antietam Creek site, the degradate, one of them was always higher than the parent compound throughout the year. And at some times, really achieving some pretty high concentrations, almost 1,000 nanograms per liter. At another site in the Susquehanna, you'd see a very different pattern where it's almost opposite. When the degradate is high, the parent compound is low, and vice versa. And so these are some of the complex issues that need to be teased out, and again, identifying data gaps. So this paper demonstrated, as do numerous papers that we evaluated, the importance of considering complex mixtures, importance of flow and other environmental factors, and the need to look at some of these degradates. So I am now going to pass it on to Greg, who's going to talk about one of his favorite papers.
All right, thank you, Vicki. So I'm going to just touch on another example here. This is Hartwell 2011. And the conclusion in this paper that was so interesting is that we know that the profile of active ingredients that are used on the landscape over time, and this is both in our developed landscape and agricultural lands, we know that over time, the profile of active ingredients changes. And this paper attempted to look over a period of time and using toxicity units, it normalized the amounts and the toxicity factors for known pesticides in use using the best usage data we have available. And through that toxicity unit method, reached a conclusion that while the quantity appears to be going down, if you convert to toxicity units, the overall toxicity is not going down. And so it was a novel method and a novel approach, and really helps us understand that this is a fluid issue in terms of what pesticides are in use, and understanding that helps us design our monitoring program so we can be looking for them in the environment, particularly in our surface waters. But it also shows us that it's possible that the toxicity all taken together can also change over time and maybe is not headed in a downward direction for less risk and less toxicity. And again, that was based on this novel approach of converting used pesticide amount and known toxicity to these toxicity units. So really interesting approach. And I don't think this has been redone since it was done a little over a decade ago. I personally think this is one that we should consider replicating and thinking about whether this approach and this method is something that we could repeat and see where it appears things stand now a little over a decade later. So because of that novel approach and all the way that it informs us, I think it's a really wonderful paper, and you can see even more details about it in our summary report that we're presenting to you today. So that was Hartwell 2011 example. And, we're really lucky to have Dr. Hartwell still working with us and involved in the project. And so I'm going to get to hand it over to him now for a favorite paper of his. Ian?
Yep. Can you hear me?
Yes.
Yeah, I'm looking at a paper by Brett et al. From just a couple of years ago, and they were looking at the impacts of atrazine on the microbiome of oysters. And oysters, of course, are a really important species in the bay. Early European explorers were having trouble exploring Chesapeake Bay because they kept running aground on oyster banks. But anyway, so there's currently lots of efforts going into revitalizing the oyster populations in the bay with mixed results. But in any event, one of the things that Brett looked at was the effect of atrazine on the microbial community that lives in and around the oysters themselves. What they did was they just exposed oysters in batches to various concentrations of atrazine, and then followed them for several months. And what they found was that the impact of atrazine decreases the beneficial microbial community that grows around the oysters, and also increases the pathogenic species of microbes that grow in and around the oysters. So, and it affects apparently the growth rates of the juvenile oysters by impacting the microbial community around them and in them, both around them and in their guts and inside their shells. So the thing about the atrazine is that it is the most used pesticide within the Chesapeake Bay region. 75 million pounds, that's a lot. It's been banned in some places, like in the European Union, but it's still used extensively in the United States. So the impact is not only on the disease vectors that impact the oyster beds, but also the growth rates of the animals themselves, which in turn impacts the success of replanting. And I think I turn it back now over to Vicki.
Thanks. So just to wrap up the report from this working group, some ways you all can help out. If you know of any pesticide studies that relate to the Bay or that you think are important, please send them on. Also, if you know of groups that might benefit from either a briefing by one or more of us or a copy of the report, please let us know. And lastly, if you have an interest in this and you want to join us, we would love to have you help out. And the next steps that we will be doing are, as I mentioned, we will continue and hopefully put out some sort of annual summary of important findings regarding pesticides. And then we hope that this report will be finalized and published in January. Thank you very much.
Great. Thank you all. very interesting stuff, and we're really excited about getting this report out there. I think it's going to be really helpful. So we do have time to do some Q&A with you guys, and we have a couple in the Q&A module. First one is, can you please explain toxicity units?
Yeah, well, I think Ian would be most qualified for that if he's
What would-
able. Yeah, we're asked about toxicity units, Ian, and the method that you had in the paper for calculating those toxicity units.
Yeah, we basically it's the relative toxicity of a given pesticide for its concentration. It's very simple. You just divide the toxicity by the volume of pesticide.
Okay,
Okay,
great.
yeah. So I
All
hope
right,
that
we have
answered is a calculation that is taking the volume with the toxicity factor to come up with total units. And that normalizes the data so that we can look across all those active ingredients and make some assessment of overall toxicity and risk. So I hope that answers the question, Bonnie.
Okay. All right. I think this one may have pertained to the graphs that Vicky had shown. The question is, what is the x-axis in your graphs? 3 what?
That was actually the months. So we took monthly samples, surface water samples for that. And so 3 is March. And then in April and May, we had 2. We did it every 2 weeks. So 4.5 is halfway through the month.
Great. Thank you. All right. Here's another question. Have you looked at the interacting effects of temperature and other stressors upon the toxic effects?
Yeah, that's a really wonderful question. And my personal opinion is there is not very much data out there about what we can expect from rising water temperature and air temperature with regard to toxicity and synergistic effects of we know the fact is our surface waters are something of a chemical cocktail all the time. There are multiple potentially toxic chemicals present most of the time. And the question is, what's going to happen when surface waters continue to warm? Is there any greater synergy among those contaminants? And that's one example, I think, of needed research, maybe gaps in research. I don't know of very much that's happening with regard to what does climate change say about risk to our watershed with regard to chemical contaminants. It's not much happening that I'm aware of, but there are intriguing questions around it for sure. No, if Vicky or Ian has anything else on that.
No, I mean, I agree. There are a few papers out, not necessarily in the Chesapeake, that sort of begin to address that, but it's definitely a data gap. And, there's so many things to consider with rising temperature. Are there more transformation products? Are the fish or other organisms more stressed? There's, yeah, it's a big question, a good question.
Great. Thank you both. We have another question. Has atrazine effects on freshwater mussels been studied and any papers published?
None that I'm aware of. We could certainly look for that and get a response back. I'm personally not aware of any papers on atrazine toxicity to mussels.
Thank you. All right, here's another question. Have inert materials been tested along with and separate of the active ingredient?
Well, certainly that is part of the pesticide registration process, which assesses overall risk of the product, and there are allowable and not allowable additives for our pesticide formulations. And we hope that during the registration process that is considered and recognized, and that only approved and accepted additional elements are added to our formulations in addition to the active ingredients. So I think that happens during the registration process, that those additional ingredients are reviewed. As far as their toxicity, hopefully that there's a good foundation in that review that is based on known toxicity of those additional ingredients.
Great,
But I
thank
would
you.
also say that maybe a lot of times when we're monitoring, we're not necessarily looking for those inert ingredients.
Yeah.
Good point. Thank you. Here's another question. What do you consider the most important gap in our understanding of managing exposure to pesticides in the Chesapeake watershed?
Well, we don't benefit from an extensive monitoring network on this particular set of pollutants. It's very snapshot oriented in the way that we capture data about the presence of some of these pesticide active ingredients and their degradates, so that we can all come together to help understand what that might mean. Yeah, we do not have a network. In fact, we only had a couple of long-term data sets for pesticide monitoring in the watershed, and we're not sure that those have been maintained in recent years. So I would say the largest gap, and maybe it's not surprising, you put some scientists together, usually that's a question about we would like to have more, but we really have very scarce data and nothing longitudinal, nothing long-term that we can look for trends and make some other scientific assessments like that. So that's my pick for biggest gap in understanding conditions.
I guess for me, one of the things being a fish biologist is trying to understand the complex mixtures. And it goes along with what Greg said, is that we don't have all that information to know what the organisms are being exposed to. But then we also don't know, because, it's hard to do even exposures to complex mixtures. What mixtures do you pick? How many do you do? And it gets really expensive. But I do think that is something we need a better handle on, is how do these either act synergistically, additively, antagonistically, as far as effects.
Thank you. We have another question. Are there any studies in the report looking at the concentration of neonicotinoids in the bay and their impacts? She says she's especially interested because in the last few decades, seed treatment with neonics has gradually become the norm in field crops.
Right. Well, I think we have some toxicity factors that are available to us for neonicotinoids, helps us understand more about the relative risk from that group of active ingredients. What we don't have is a lot of data on presence, or sometimes we say occurrence. We just really, I don't know of papers on neonicotinoids in surface waters in Chesapeake Bay. Now, when we get these specific questions about a particular ingredient or a particular receptor that's at risk, like the mussel question, it really requires time to go jump into the literature unless by chance we know of a paper. So it may be out there. We just aren't aware, but boy, it's probably very sparse. So I think we understand more than we did a decade or so ago about the risk that's presented by neonicotinoids, but I'm not aware of much data on occurrence in surface waters. So that may be another data gap that we can identify. We can go look for papers, see if they exist, if there are any investigators out there doing that work. And if not, the question is that a high priority issue for us? And if so, how can we come together to get more data and do some monitoring and testing? And that's really, I think, an example of how this project was designed to work. We look for where there's data, we look for where there's gaps, and from that we make some decisions about how we can add value. And maybe that's one. So anyway, that's a response on that specific question.
Great. Thank you, Greg. We do have time for probably one more question. Do fish species differ in their response to pesticide exposure?
Hand that to our fisheries biologist.
Yes, the answer is yes. I mean, we find a lot of difference between species, not just even in responses to pesticides, but other chemicals as well. And some of it depends on their physiology. Some of it depends on their sort of life history, like do they spend more time close to the sediment? Do they spend more time up in the water column? You know, what are they eating? All those kinds of things go into it. But yes, we see big differences in responses.
Wonderful. Thank you. These are really great questions. I'm going to take these questions back to our research working group so we can continue to talk about some of the things that you all were interested in. And I thank you very much for that. So I think we're ready to make a transition. So I'm going to turn things back over to Greg with my final thank you to our distinguished panelists for your presentation today.
Yes, thanks for a chance to update everyone on our work. And thank you again, Ian and Vicki. So now, without further delay, we're going to get to our keynote and dive real directly into our theme for the conference. And I'm very honored here to introduce Dr. Elaine Ingham and Dr. Adam Cobb. Dr. Ingham is founder and president of Soil Food Web Incorporated and director of the Soil Food Web School. Dr. Ingham uncovered the soil food web nearly 4 decades ago and has been pioneering research in the soil food web ever since. Widely recognized as the world's foremost soil biologist, she's passionate about empowering people to bring the soils in their communities back to life. Dr. Elaine's soil food web approach has been used to successfully restore the ecological function of soils on more than 5 million acres of farmland all over the world. The courses offered by Dr. Elaine's Soil Food Web School have been designed for people with or without a science background, making them accessible to individuals who wish to learn and to begin a meaningful and impactful career in an area that will help to secure the survival of humans and other species. And I'll introduce Dr. Cobb next, but let's go ahead, Dr. Ingham, and hear from you.
Hi, we're really excited to be bringing some ideas, and the topic that we're really going to focus on today, Dr. Elaine and myself, is soil life being our future.
We teach people how to make compost, how to apply compost, how to determine whether your soil is being built or whether the organisms in the, your soil are dying out, and pretty soon you're going to have dirt instead of soil.
My name is Dr. Adam Cobb, by the way, to the audience, and I am the science communicator here at the school, which means I get to look across all of our programming and say, how can we really help people get it? How can we really help them take these complex ecological, biological realities and have systems that are really regenerative, that are bringing the soil back to life and reducing the need for any of these costly and harmful inputs? So you mentioned dirt and soil already, Elaine. That's where we always open in these things, right? A lot of modern agricultural practices utilizing heavy doses of chemicals are actually making dirt. And we're trying to help bring forth this new way of regenerating soil and treating soil as a living ecosystem. Dirt is really just the minerals, the lifeless component to us. Dirt is on the bottom of your shoes, right? And then when we talk about soil, you take those minerals, but you add organic matter, and then those especially aerobic organisms, the life in the soil. And then it's not just some dead, lifeless thing, it's a thriving ecosystem. That's what we want to achieve.
And it's going to benefit all of the plants. They are going to have higher amounts of nutrition in their bodies so that when you eat or your animals eat that material, you're getting the proper sets of nutrients in the proper form for you to be able to take up, for your animals to take up. So the organic matter is really the food to feed the aerobic organisms, the bacteria, the fungi, the protozoa, the nematodes that should be in your soil. So, now adjusting for the growth of different kinds of plants, you can select 4 weeds, which most people are not going to want to do, really. So we want to make certain you understand how to suppress the weeds and get more of the crop plant that you desire, get more of that growing, higher levels of nutrients, better flavors, a whole list of things that we'll go through later.
Absolutely. And speaking of dirt, I think it's important for everyone to understand how we got to where we are, where a lot of the research coming out of institutions, agricultural institutions, large agribusiness, is really based on what I would call dirt farming, right? How to manage these systems with chemicals when all of the life has already been, or not all of the life, but
the
most of
beneficial
really,
life.
yeah.
Yeah, the beneficial life is no longer there. And what you've got are diseases and pests and problem organisms. And now how are you going to switch things around and exit the production of harmful inputs? And, one of the big things is you've got to stop tilling because every time you till your soil, you're wiping out 50% of the beneficial organisms. And now the plants don't have their buddies to help them grow into the soil. Very easy to compact where you've fluffed up your soil doing tillage, starts to rain, you don't have any protection on that soil surface, and you're going to have compaction. And that means that the roots of your plants won't be able to grow down to the summer water. That if it's soil, that's what a major component of these organisms do for your plant, is to build the structure that holds the water so you don't have to irrigate. Could you save yourself a little money if you don't irrigate? But you do have to have the soil that's not compacted. You want to have the roots of your plants going down as deep as they possibly can.
Absolutely. And to avoid these harmful inputs that reduce the functions and the abundance and the diversity of the soil life, but also end up in our water systems downstream when we have these chemical agriculture methods going on upstream. Excellent. So this is one of my favorite bits of evidence to bring to the table. You can see the article that this came out of, calculations on what we would call the relative biomass of life on planet Earth. And it is truly a planet that is dominated by plants. Humans proportionally are a tiny, tiny percentage of the weight of life on Earth, even all the animals taken together. And look at the microbes, the second largest category. They realized millions of years ago how to work with plants to achieve what they want. They got fed by the plants. They received safe places to live near the root systems of the plants. And some microorganisms cheated or learned to become diseases to plants, but the vast majority are out there that are beneficial, which is very different from the conversations that professors were having with you in grad school, right, Elaine?
I would listen to various versions of it's absolutely a very bad thing. Nobody has any interest in the organisms in the soil because those organisms don't do anything. They do nothing to help your plant. Most of the time we're spraying toxic chemicals to try to get rid of those organisms. And as I went through and did my work, the total opposite is reality. In areas where there is no diseases, no pests, no problems, no weeds, on and on, we have very good growth of the plants, and the biology abounds in that soil because there's all these protective things that these microbes absolutely do for your plant. And so trying to understand that, and how do we figure out which are the good guys and which are the bad guys? So, if you're interested in those sorts of things, come work with us.
There you go. And that leads us right to this, which, I haven't done as much of the labeling of this food web diagram. And in fact, I think everybody should know it's many, many times more complex than this. This is a simplified version. But the importance here is that we recognize the power of the relationships in the soil, right? It starts with the sun and the plant's relationship to the sunlight energy, where it can make sugars through photosynthesis, and then it can share those sugars with the beneficial organisms in its root system, the fungi, the bacteria. Then there's predators that come in, on and on and on through these levels of complex interaction. And in our next couple slides, we'll summarize some of these benefits. But to me, I always want to come back to this. All those little arrows and colors and things are to let us understand something of how complex the story of soil really should be.
When these organisms take the nutrients out of the sand, silt, clay, rocks, and pebbles, that's an inert form. Plants can't take up the nutrients in rocks and stones and pebbles, and so you've got to have the biology. The more biology we can get back in, the more nutrients your plant will have, the more things that these plants can protect themselves from.
There we go. And that is summarized here on this fairly busy slide. But you have some pictures here of fungi and nematode that are part of this this process. I like to summarize that the bacteria and fungi, as you said, they're pulling nutrients into their own bodies. They're also building structure in the soil, stability in the system. Especially the fungi can make large soil crumbs or aggregates because they're winding around with their filaments as they're growing. And then you need those protozoa, nematodes, and some of the insects to come along and prune the forest a little bit and release the nutrients back into the system. And they also, protozoa and nematodes, become, for us, really important indicators of the system's health. If they're missing, something's wrong.
If the only thing you've got in your soil is bacteria, you can probably bet that those are the not so wonderful bacteria. And so we teach people to be able to determine these kinds of organisms, one from the other, and what the biomass is. Are they doing what they're supposed to be doing, or are they bad guys and doing things that are very unproductive for your plant? So we teach you what these organisms look like. There are both bacteria and fungi. Those long strands are the fungi. Look down into in the next one, that's a nematode, going in after some bacteria to keep itself alive. And in return, it releases nutrients into the soil that your plant can obtain.
Absolutely. And these other essential biological functions that only happen when you have diverse and abundant bacteria, fungi, protozoa, and nematodes. First and foremost to me is the protection of our plants, the suppression of diseases. You can always get an organism that floats in on the breeze where you live, and that could be something that would attack your plants. But if there's competition from all of this other life, inhibition of the disease, consumption of the disease-causing organism, these can all be dynamic form of stress reduction. And then there's all that nutrition to the plant, so the plant can have a strong immune system as well. I like to use this metaphor of an arch to explain the mindset shift. So a lot of people get focused on learning about vegetation management or landscape management, which are important. They're pieces of the arch, but if we're missing the keystone of the arch, it's not going to be stable. It's not going to work. The keystone is soil life, and any system that is not designed around the functions of living creatures in the soil is going to ultimately be fragile. Here's a great example of where we can do this comparison. And Elaine, you've seen this time and time again in the world when people have small plants that are struggling and hitting compaction layers. It's just dirt and they're using chemicals. What are those outcomes?
You're going to have to apply really toxic materials to try to get the plant to grow. So inorganic fertilizers are nothing but a salt, basically. Lots of different ways you can arrange the molecules there, but the basic problem is that it takes water away from those inorganic fertilizers take the water away from your plant as well as the microorganisms in the soil. And think about a bacterium, fungi, protozoa, nematodes, things like that. They're so much smaller than the plant that they can't kind of live through a period of drought the way your plant might be able to do that, especially if we can get the root system going down deep into the ground, build the soil structure so there's oxygen all the time, that deep, then there's not a problem. So we've got to make certain that we make the conditions favorable to the growth of the beneficial organisms. That means your soil will stay in an aerobic condition. So oxygen can come down from above the surface of the soil, be moved throughout that whole area. And those conditions are very disease suppressive. We see the same thing with weeds. Weeds love to grow where there's a problem with compaction, where there's too much water puddling in the surface because the water can't move through the compacted layer, that black layer you see over on the left-hand side. So which system do you want? Do you want to have to pay all of the inorganic fertilizer money and getting that down onto the soil surface. And now you're going to have diseases and pests and problem organisms. These the dirt conditions are selecting for those weeds instead of the crop that you want to harvest down the line. And you can see why it is much more expensive to be using the chemical approach. Instead, let's use Mother Nature. You really only have to make compost, good compost made properly with the right levels of bacteria, fungi, protozoa, nematodes, microarthropods, earthworms, all of those things need to be in your soil, build the structure. So you have the nutrients the plant needs, you have the water, you have protection against the diseases and pests. And oh yeah, above ground, we want to have those same microorganisms covering all of the leaf surface, the bottom of the leaf, the flowers, the fruit, the stems should all be completely covered with these beneficial organisms. Then when a disease-causing spore of some kind of disease-causing organism, when that spore or the organism itself lands on the surface of your plant, your crop plant, there is no way for that spore to germinate and start to grow and get inside the surfaces of that plant, attack and start destroying your flower or your seeds or the underside of that plant. So we want to make certain that all these beneficial organisms are present, and you don't have to pay for pesticides or insecticides, or think of all the things we spray our dirt for. So we want to work with nature, and we want to teach everybody on this planet how to work with nature and not have to buy any of these toxic chemicals.
So I think in summary, we can talk about dirt as a loss of profits, right? When people are dealing with compaction layers and the root systems of those plants are not able to grow deep and take advantage of the nutrients and the water that is in deeper soil layers, you're going to have to spend more money on irrigation or nutrient inputs, and you're also going to have reduced biological functions in dirt. When those organisms are missing, then you're more likely to have issues when drought comes along. You're going to have poor nutritional value in what is produced from that system. And it's going to ultimately, you're going to be chasing problem after problem after problem and trying to kill problem after problem after problem instead of investing in the structure and stability of this system. So soil is the solution. If dirt is the problem, soil is the solution, because when you actually have thriving soil life, you can have reduced stress on the plant, you can overcome compaction, you can avoid erosion and the runoff of these toxic materials into local water sources. And in this picture here, Lane, with the Swiss chard that these students are growing, you told me that sort of where they've got those little red flags and anything closer in the foreground of the photo, they've actually been putting liquid amendments on.
This is actually just one application of the compost extract on the surface. And they tried to spray underneath as well. You can see going further out in the opposite direction, you can see that those Swiss chard are not doing very well. There's a place where they're almost been wiped out by insect pests. That's how close the bad guys are to our area where we applied the compost extract. And the chard was able to overcome the attack by the aphids, and the aphids either died or went back to the other row and part of the row because they didn't have to fight. They didn't have to, they're not competing. They don't have to deal with inhibition of their growth rate by the beneficial organisms. Whereas on, the other side with the proper sets of microorganisms in the soil, it's a no-brainer. They don't have to fight those battles because the microorganisms will be doing it for them.
Right. Absolutely. Okay, so this is a summary slide of a lot of what we've said, that we're utilizing the microscope as a tool, a very important one. And so at the school, we teach people practical microscopy so they can see who's there, who's missing, are there diseases, do you have the diversity and abundance of the beneficial organisms, etc. And then effective bioamendments comes down to the compost and the liquid amendments that are actually full of living organisms, right? So we're not just putting nutrients out there, we're putting life out there, letting it reconnect with our plants. And then there is another part of this, which is what I call holistic agroecology, that we're not doing the things in our land preparation management that would set life back. So you mentioned tillage earlier. Chop up the soil all the time, you're killing especially your fungi and in huge rates. But I mean, even the nematodes and protozoa, I say, are quite sensitive to land practices that are not designed to help them achieve health and resilience, right? We have to look out for these little guys, and then they'll look out for us and our plants. So that takes us to something that people may be really excited to learn, that it can be hard to align these things in the beginning, but once you do it, once you figure out the tools to bring to your property, to your garden, farm, ranch, etc., regenerating the soil actually creates what I could call like a virtuous cycle. All these benefits come into play. You have to push that boulder up over the hill using microscopy, biological amendments, holistic agroecology. You have to change the ecosystem's state. And when you get those things aligned and the soil starts to come back to life, it gets easier.
We work with nature, not against nature.
Excellent. Okay, we're in the last few slides that we're going to present today, and it's just a summary of some of the key benefits. Like once you get this soil ecosystem thriving again, once you make these investments in soil life and you get from dirt back to soil, what can we expect? What can we expect that system to do for us? What do you think about benefit number one there?
It's really important to have structured soil so you can get the oxygen moving down into the soil and you never become anaerobic, and that never, those anaerobic diseases and pests and problems, the weeds aren't going to bother you. So we want to grow those deeper roots. We want to have the organisms growing at every step along the way.
Excellent. And along with that is the fact that we can store more carbon below ground, right? When we get those roots going deeper, and especially when we get the fungi and bacteria going in that community going in the right direction, in abundance and diversity, we see the accumulation of carbon in these systems, which just makes everything easier. It buffers the entire system from problems and leads us to a place where we're building up the soil every year instead of degrading it.
Yep, and it's increasing the food materials that the organisms have to grow on, and increase stability all the time by more and more diversity for both the bacteria and fungi, leading to protozoa, nematodes, etc. So here is retaining and cycling nutrients. We've talked about this a bit. We've got to remember that the plant requires soluble nutrients. Somebody else needs to have done the work, and then the plant can take it up.
Benefit number 4 goes with the nutrient cycling. It's the health of the plant, right? So its immune system is functioning properly because it has the nutrients that it needs. There's also, you referred to this earlier, like what I call microbial armor. All the surfaces of the plant have beneficial organisms on them. They're glued to the above ground surfaces. They're acting like the stomach of the plant below ground and helping to digest the world that they find themselves in, the soil world. But it's the fact that there's armor there, that anything that could cause harm to the plant is going to hit a barrier of the life that we actually want.
Benefit number 5, we've talked about this before. We want to have the biology that's selecting for those organisms that would go after a grasshopper like this. So you don't have those outbreaks of the predators further up the food chain. So we want to make certain that we don't allow the conditions to be such that it would select for the weeds. Instead, make certain you're selecting for those other bacteria, fungi, protozoa, nematodes, microarthropods, earthworms, that they're all going to be taking these guys out long before it becomes a plague.
Benefit number 6, I think, will be very exciting to this group, because with the Chesapeake Bay right there, I know that there's a constant issue with water quality and pollutants. So when we actually get a soil ecosystem going in the right direction and the life is there and the life is thriving, it can help decontaminate our environment, not just on the farm, but downstream of the farm. You're not having all of these excess nutrients and pesticidal compounds accumulate in your water sources. So this is a huge benefit as we need to clean up our world, as we need to partner with life to do that essential work. And this scales all the way up, if we really get wild with it, to think about planetary scale and human civilization scale resilience, right? The ability to overcome stressors. When we have systems that are based on soil life, they put us in the position to regenerate all of our biosphere, the entirety of planet Earth. And a lot of that is because we also stop the constant damage coming out of these chemically intensive systems. I think our last slide here, Elaine, I should kick over to you to let people know about how they can take another step in their understanding of these principles.
Yeah, we want people to, first of all, start at the beginning of the foundation courses, where we give you enough of the background information that you'll need to grow a crop using biologically friendly approaches. Some people come to us where they've never grown anything at all in their entire life, and that takes a little bit of time to get you coming along on that, but we will have that, we'll be able to recommend places to go or people to talk to after we gauge how much help you need. So all the principles that we've been talking about are gone over when you take the introduction to the soil food web. So FC2, foundation course number 2, is really about making compost, making the liquid amendments. We teach you that in FC3, foundation course 3, and then we do a course on microscopy, because this is the easy, simple way. After just a few minutes, that's all it takes. You go out into the into your place you want to know what the biology is. Has it increased since last time? Has it decreased since the last time you applied it? What's going on? We've got to find that out. And these are simple and easy ways. We can teach you how to use the microscope in a day. We teach you how to interpret what you're seeing using the microscope. After a couple of weeks, you should be getting a really good idea of how to do that interpretation and how do you make reports to clients that you might be working with. So the basics are covered here. Now we also have other courses, advanced courses, if you will, where we go in a lot deeper, where you need to have gone through the foundation courses and have a knowledge of how does my plant grow? How do I need it to be growing? How can I improve it? So all aspects of the the desire to have plants that were raised with the biological approach. We're working with nature, not against. Okay, so I think I hope everything was clear from that introduction. I think we probably have some questions here, though.
Yes, absolutely. And just before we do that, I didn't get a chance to introduce Dr. Cobb. So let me just do that real quickly so we get to know Adam Cobb a little better. His passion for agroecology emerged 15 years ago while volunteering on farms in New Zealand. He spent several years as a postdoc researcher and university instructor. He's taught courses on food security, restoration, ecology, and environmental science. Adams contributed to more than 20 research publications linking plant microbial symbiosis with enhanced nutrition and holistic soil health. And he joined the Soil Food Web School in 2021, and he's following his dream to help regenerate soils, improve human nutrition, and enjoy the splendor of our living planet. So I wanted to be sure that we got to know Adam through that introduction. So thank you for giving me a minute for that. Thank you so much, Dr. Ingham and Dr. Cobb. And yes, we do have some questions here. In fact, quite a few. I'll ask you a couple that we have time for. And if you're able to continue in Q&A to answer other questions, we really appreciate that. So one question came in about special impacts or effects when soil amendments are derived from sources that have human microbial matter in it. So thinking here about municipal biosolids, for example, any special impact to the microbiome when those soil amendments are used?
Well, they're going to have to first understand in making compost, you never allow it to go anaerobic in any way that you can possibly manage. And most municipal composting, see, I'm having troubles with that whole concept, sticking them together, because those, and if you've ever driven by the local, compost producing area, it stinks to high heaven. And that means it's anaerobic. That is not compost. You're going to kill your plant if you put your plant into something like that, because it's got so many toxic materials. It's got so many disease-causing organisms. They've won. So we have to get people understanding that making compost is a really important part of this, because then you'll get organic matter that is maintained by those organisms that are going to be aerobic, where everything we do has to select for the beneficial organisms and against the diseases and the pests and the problems. So yeah, let's not confuse people by calling waste material compost.
Hmm. Okay, very interesting. Wow. Okay, so a next question, and I imagine this is one that comes up frequently, and it is about scale. And while we appreciate the benefits of this, we've got a lot of mouths to feed in the world. So how do we envision this with regard to food security and scale?
Well, we
Ooh, I'd love to
produce.
take that one.
Okay, you take it.
Okay.
All yours.
So a reference I'm going to add in chat is just our YouTube channel, where we have very large scale producers as guests of our webinars often. And people actually at various scales, right? There's farm to market gardeners all the way up to people doing commodity crop production on 100,000 acres, right? So it is very scalable. And in fact, we get reports often from the large scale producers, ranchers, and farmers, many of whom are friends of mine, that they have saved millions of dollars by switching over to what I would call biological soil management in place of those expensive chemical inputs, right? It's not magic. There's nutrients to manage. There's all these other things to consider in the system. But if you realign the system, and really it starts with realigning our mindset around the living soil, the reports are in from many sectors, and increasingly the scientific literature is also showing that these methods are optimizing profits, and not even through just reducing yields. That's a lot of times people, I think large agribusiness may be part of this, Elaine. They look at all this research and they say, you can't feed the world this way. And I say, well, what are the assumptions based on? Right? What are they looking at? Is it all based on dirt farming? Because I studied arbuscular mycorrhizal fungi in grad school for years and as a postdoc. And if you put them in dirt, they don't do so hot. But if you put them into a living soil ecosystem, they can really help plants to produce a lot more biomass and food quality.
Okay, thank you for that answer. And there's reason for hope there for sure. Okay, thank you. Here's another question. Are there any differences between the composting approach that you teach and the standards required to maintain USDA organic certification?
There's quite a bit of difference because they've really, the USDA certification has focused in on a couple of things that aren't really that important. The most important things are you've got to keep it aerobic, because most disease-causing organisms, not all, but most require anaerobic conditions in order to outcompete the aerobic organisms. So that we've got to make sure we are promoting the beneficial things. So we'll have the compost that we want. We'll have the organisms that we want. And when you start looking at the number of bacterial species in a compost, or fungal species, or any one of the groups that we've talked about, it's people are estimating that we've got a couple thousand bacterial species per gram. In actuality, most of them aren't active and doing their thing right now, but shift the temperature by 5 degrees. The ones that were in there, they're gone down, functioning in that compost, doing the proper things. They're going to go to sleep. But then the next set of microorganisms that are promoted by that colder temperature or higher temperature, more water, less water, all of these things we think of as being associated with biomes or ecosystems. We've got to have that diversity of microorganisms so that the organisms are constantly doing the process, preventing the diseases, the pests, the problem organisms. Adam, do you have anything to add to that?
Yeah, I think that USDA certification has really focused on heat, X number of days. You have to turn the windrow or pile X number of times. There's a lot of boxes to tick, but the main mechanism is considered to be heating the microorganisms to the right threshold, heating the material to the right threshold so that you knock back most of your pathogens, right? And we add another aspect to that, which is the biological competition, right? The inhibition of the pathogen, the consumption of the pathogen, the direct competition for resources. And then even as you put these composts on your plant, by improving the nutrient cycling, you upregulate the immune system of the plant. And I think that that is a better bet, because oomycetes float around in the air and can cause huge problems on farms if they don't have competition or if they land in the wrong environment. Lots of things can come in on the feet of birds to your production system. So instead of thinking about sterilizing the material, we need to think about getting a thriving community that will maintain itself and prevent the diseases from taking hold. But there's some work to be done with USDA organic certification. There's a few trade-offs that are necessary if you go that way. It doesn't mean we can't fix your compost on the back end with worms or something else that will really improve the biological community. But any of your certified producers need to be aware of the extra hoops they have to jump through by the regulators.
Wow. Okay.
Yeah, so we've really done away with the need for anybody to apply pesticides of any kind. You don't need herbicides. You don't need all of these acidal things to kill, is what that aside means.
Mhm.
And so use the proper organisms. And once you get them established in your soil, they're not gonna dissipate. They're not gonna be killed by winter or by a little bit of dryness or a little bit of whatever. You have something in perpetuity that's going to do what you want without costing you any money at all, because the organisms do the work for you as growers.
Okay. Well, thank you for all of this. I think we are just up on our time here. So again, thank you so much, Elaine Ingham and Adam Cobb. There are lots of questions in Q&A, lots more we wish we had time to talk about, but if you could do some work in there and provide some answers, that would be most appreciated. So thank you so much for joining us today. And we are now going to switch over to another chat question. Right, so we're proposing this question to you all. Actually, it's two questions. What do you think are the impediments to achieving healthy soils? And how can we better support transitioning to healthy soils? This set of questions in particular, we're hoping that we'll get some feedback and information that will inform our work groups. Each year at the conference, we try to ask some questions and get some discussion going that will help us consider what the high priority items are that we should be working on. And so we'll be capturing the result of this chat. We will save it. We're going to save the entire chat and send it back out to participants. So you will get to see everything that has been added to chat today. And again, we will use it in the project to help inform our work groups on priority issues. So asking you to put your thoughts in chat with regard to these questions, and take this time maybe to stand up and stretch or take a very quick break. We're only going to do this for 5 minutes, but we'd like to get your thoughts and have you take a stretch during that 5 minutes. So we will be back in 5 minutes. We'll see what we've got in chat a little bit and then go on to our next fabulous presentation. See you in 5 minutes.
Welcome back, everyone. I just want to acknowledge, if you stepped away from your screen, that we are so very grateful to our conference sponsors, and we hope you'll get to know them all. Mom's Organic Market, Cottingham Farm, Foxhaven Farm and Learning Center, KW Landscaping, and Clean Water Action, as well as our generous funders listed on the previous slide. Thanks to all of you. And I'll turn things back over to Greg.
Okay, thanks, Bonnie. And thank you, everyone who's been putting some thoughts into chat related to these questions. I guess what I take from what I see there so far is the impediment is us, and maybe we knew that to begin with, but some hopeful suggestions in here. While we recognize that this is a movement that would have a wide range of benefits, people are recommending things like further research and education to help make the case that it is scalable and it is something that can be done at a scale that'll bring these many potential benefits to our environment. So while we know it's a social science question, I think that's maybe another conclusion I draw from this, is that maybe we need to know what the footprint of social sciences is in our project here, and whether we're really using that wisdom of practice to help drive behavior change. But we do see some hope here. So yeah, and if Dr. Ingham is still with us, and Dr. Cobb, there are a few comments in there that might, again, be right up your alley, and you might be able to provide some more information around. Again, we're going to capture this chat record and refer back to it, see where that might lead us with some future initiatives. All right, well, next we are going to bring our next presenter to the fore. Our next presentation is by Dr. Kendra Klein. Dr. Klein is Deputy Director for Science at Friends of the Earth, where she leads work on food and farming solutions. She's a writer, researcher, and advocate in environmental sustainability, food, agriculture, and environmental health. Before joining Friends of the Earth, Kendra coordinated farm to institution work at the California Healthy Food in Healthcare campaign. And she worked at Breast Cancer Action on chemical policy reform and corporate accountability related to pink ribbon fundraising. Kendra has apprenticed on organic farms in California and Hawaii. She is a 2011 Switzer Environmental Fellow and has written for The Guardian, The Nation, Gastronomica, Civil Eats, Food Tank, and EcoWatch, as well as multiple peer-reviewed journals. She has a BA in Interdisciplinary Studies from Miami University of Ohio, and a PhD in Environmental Science, Policy, and Management from UC Berkeley. So now, please, we will hear from Kendra Klein.
Good morning. I'm so glad to be joining you today. I am going to share information with you about a recent report that Friends of the Earth published on genetically engineered soil microbes and some of the risks and concerns that we have related to them. Soil is one of the most biodiverse ecosystems on the planet and also one of the least understood. We are just beginning to scratch the surface of our understanding of the complexity of soil ecosystems. In just a teaspoon of healthy soil, there are more microbes than there are people on the earth. And in a handful, there's more biodiversity in just the bacterial community than you would find in all of the animals of the Amazon basin. We have a lot to thank the micro and macro organisms of the soil for. If you like to eat, thank these organisms, because over 90% of the food that we eat comes from the soil. And as you can see, they produce all of these benefits, benefits for us, right? Going about their daily business. Soil structure and carbon sequestration, breaking down organic matter to cycle nutrients in the soil, controlling pests, providing pollination, water filtration, all sorts of important activities. The way we farm significantly shapes soil microbiomes. And you can see there are layers of interacting microbiomes in any ecosystem, but also on farms. And our choices about whether to use compost versus synthetic nitrogen fertilizer, whether to use pesticides or not, type of tillage, whether we grow in monocultures or use diversified farming systems, all of those significantly shape the soil microbiome. Farmers, since ancient history, have been working with the soil microbiome. Using compost is a way to feed the microbes that you want to generate rich, healthy, living soil. More recently, farmers have actually applied microbial products, and these are known as biologicals. So any agricultural product that's derived from a living organism or plant. And this is in stark contrast to the majority of inputs used today in agriculture, which are derived from fossil fuels. So microbes, pheromones, biomolecules like amino acids, and these can be used as biostimulants, biofertilizers, and biocontrols or biopesticides. These have been used for a long time. 1938 was the first recorded time that a microbial pesticide was sold to a farmer. BT is still a very commonly used microbial pesticide. Distinct from BT genetically engineered crops, applying BT is approved for organic production, as are most of these microbial products. Today, there are over 1,200 different companies worldwide that offer many hundreds of different biological products. We are in the midst of a major boom in the biologicals market. The global market is expected to nearly triple in a span of 8 years and reach 29.3 billion by 2029. And you can see the fastest growth is in biopesticides. This summer, I was at the Western Growers' first ever biologicals summit, and that gives you some indication of the fact that biologicals are really going mainstream. Western Growers is one of the largest agricultural trade groups in the US. They cover veggies and fruits and nuts. And the conversation was about this growing market. A lot of motivation voiced was because consumers are demanding food that doesn't have toxic pesticide residues. So it's interesting that biopesticides is the fastest growing part of this market. We've seen a great deal of consolidation in the past 10 years. So these are the largest agrochemical companies, and you can see that they are moving rapidly into this space. They are buying up traditional biologicals companies, along with companies that are giving them the capacity to genetically engineer microbes. All of these companies are offering biological products. Many of them to date are not genetically engineered. So we have to keep this in mind when we talk about biologicals and microbes in agriculture. Many of them are not engineered, but we are likely to see a real increase in the number of engineered microbes offered. And because of that trend, Friends of the Earth thought it was important to compile what we know so far and to raise some potential concerns so that we can better inform the public and growers and policymakers so that we can be making better decisions about how to regulate GE microbes in agriculture. So researchers have for decades been genetically engineering microbes. For agricultural purposes, they've looked at bacteria, viruses, and fungi. Bacteria are by far the most common. The first GE microbe in agriculture was field tested in the 1980s. Some of you might remember ice minus. The presence of the Pseudomonas bacteria, this particular Pseudomonas on crops, allows frost to form at higher temperatures than normal, so increasing frost damage. And so the developers genetically engineered the bacteria to remove its ability to form ice crystals. Of note, this was not commercialized. They moved on to naturally occurring mutant strain with the same benefits, and that is actually used on a variety of crops today. And this is an important side note because from the researchers that I talked to in the traditional biological space, they raised the possibility that there's really no need for genetic engineering because there are so many naturally occurring microbes that we have not yet even identified that likely we could find one to achieve any agricultural goal we would have without the engineering. So interesting that that happened with this first one here. There are two products that are currently on the market that we know of. One is from a startup company called Pivot Bio. The product is called Proven, and it's bacteria that have been genetically engineered to eliminate the off switch on the microbe's process for fixing nitrogen. So these are bacteria that naturally fix nitrogen. And if they are in a nitrogen-rich environment, they will down-regulate that fixation naturally. What this company has done is eliminate their ability to down-regulate the production of nitrogen. The idea being that they will continuously pump out nitrogen and that therefore this product could be used to replace synthetic nitrogen fertilizer. That is a very important environmental goal. We know that reducing and eliminating the use of synthetic nitrogen fertilizer. It's associated with massive greenhouse gas emissions, with very significant water pollution. And yet this product, although it's been grown on over 3 million acres and bold headlines claim its success, has not actually demonstrated success in peer-reviewed research to replace synthetic nitrogen fertilizer. So that is one product that's out there. Another is from BASF, actually originally developed by Bayer, but they were forced to sell it off to BASF along with another set of products. And this is a 2.0 version of their Plancho Votiva seed treatment. This is a neonic seed treatment, combines a neonic, a non-engineered nematicide, and now the 2.0. This is a genetically engineered bacteria that the goal is to increase microbial activity in the soil. And they've achieved that by inserting a foreign protein that produces an enzyme that aids in converting dead plant residues in the soil to sugars. This is an activity that is happening in abundance in healthy soils naturally that are managed under ecological conditions. So this is a pretty reductionist approach to improving soil health. But BASF is using the marketing of a long-held understanding that healthy soil produces healthy plants, which are better able to resist pests and diseases. And so they're marketing this as a sort of novel, yet very old approach, but novel approach to plant health. We actually don't know if there are other products that have been commercialized, and this is because of the extreme lack of transparency on the part of our regulatory agencies. the EPA's website does state that it has registered 8 GE microbes as pesticides, but it is nearly impossible to find out what they are or whether they've been commercialized. And this image you see is an excerpt of a letter from Pivot Bio to USDA in 2020. And this is as it appears in the public record. There are 4 pages of a completely blank table. And in the margins, everything is noted CBI, confidential business information. So companies are able to redact nearly all information, including what the microbe is here, from public view. And so this completely undermines our ability as a public and as policymakers to really understand what's happening in this space and to develop science-based regulations. So risks and concerns associated with this novel technology. I want to start just with an understanding of the limits of our knowledge and the potential unintended consequences that we already know happen related to genetic engineering in the genomes of organisms. So just to illustrate the complexity and the sort of limits of our understanding of what we're doing, even when we might achieve our goals. Pivot Bio's patent for proven lists 29 different genes and myriad proteins that are involved in nitrogen fixation. And their own published research shows that they were surprised that their particular intervention functioned to turn off or to eliminate that bacteria's ability to downregulate nitrogen fixation. It just as easily could have increased its nitrogen fixation. So that we can tinker with these genetic processes and produce some outcomes does not mean that we really understand what's happening here, nor do we understand the potential unintended consequences for those organisms. We know, and an increasing amount of research shows, that genetic engineering, including gene editing techniques that are claimed to be more precise, like CRISPR, can result in an array of unintended genetic consequences. Inserting and deleting genes, inversions, and translocations that were not expected, right? So it's a bit of the sorcerer's apprentice here, where we've got enough knowledge to accomplish some goals, but not to really understand what might happen as a result. Thinking ecologically, this is very different from genetically engineered crops, which has been to date the realm in which genetic engineering has been deployed in agriculture. The scale of release is far greater and the odds of containment far smaller. So consider that there are about 3 trillion corn plants grown in the United States. Most of them are genetically engineered, and an application of GE bacteria would release the same number of modified organisms about every half an acre. This is concerning also because microbes can readily transfer genetic material between organisms and even across unrelated organisms in a process called horizontal gene transfer. And this means that the genetic modifications released inside GE microbes could move across species boundaries in unpredictable ways. And they could also move geographically very quickly. Microbes can travel on winds around the planet. And so there's really zero possibility of containment once GE microbes are out in the environment. There are also concerns related to altering microbial communities. We know that changes in the microbiome, in the soil microbiome, can result in changes to plant growth and insect communities. This is why farmers use some biological products, because they can result in changes that we do want. But they can also result in unintended changes and detrimental changes. There is a risk of introduced microbes becoming invasive species. And also, just stepping back and thinking about how we are deploying GE microbes within industrial agricultural paradigms and what that means for their consequences. So So we also need to think about the fact that because of the entry of these major agrochemical companies into this space, that genetically engineered soil microbes are most likely going to be deployed within an industrial agricultural paradigm. That's what we're already seeing with the 2 products that are on the market. These are being grown within, being used within massive monoculture, corn and other commodity crops. And there's no reason to assume that if we're using GE microbes as pest control, that they are not going to contribute to the pesticide treadmill that we're already on. When we're using them across acres of monocultures, insects and weeds are going to very rapidly develop resistance. So really we need to be shifting from this industrial paradigm to agroecological production. We have decades of science showing that that's the direction we need to go. And it's concerning that we've got yet another technology that's being developed and deployed within this very reductionist industrial approach to agriculture. There are concerns about potential human health risks, very small risk, but one we have to take seriously of a microbe being a novel human pathogen. And considering also that we are deploying them in production systems that use high rates of pesticides that we know can result in immunosuppression. And so that's a population of people that might be more susceptible to opportunistic pathogens. We have a lot of questions around consumer exposure and the human gut microbiome. We are increasingly understanding the connection between the gut microbiome and our health. And really, this is a space with questions that need to be answered. Could these genetically engineered microbes be problematic to the human gut? We can assume that people will be exposed to some amount of them on food. And then antibiotic resistance. So genetically engineered organisms often include antibiotic resistant markers in order to distinguish the engineered organism from non-engineered. That to date has not shown to result in antibiotic resistance in humans, but that may change if we are engineering microbes. And so one illustration of this is a fungus that is used as biocontrol, a non-engineered fungus, infecting a stink bug, as we want it to do to control pests. But here's also a case of it infecting a human eye. So very uncommon, but again, we need to take this possibility seriously. So this was a farmer with underlying conditions. This infection was susceptible to only a couple antibiotics. And had it been a genetically engineered fungus expressing antibiotic resistance markers, the outcome may have been affected. And finally, socioeconomic risks. So corporations will have intellectual property rights over these engineered microbes. And as we know from the history of genetically engineered crops, corporations have enforced their intellectual property in incredibly aggressive ways that have disenfranchised small scale and family farmers. And so we don't know how this will play out, but history tells us to be very concerned about the type of power that corporations are going to wield. As we talked about, there's massive industry consolidation in this space, and that will shape innovation. On the next slide, I'll give a little snapshot of that. But also just to raise that we need to grapple with the fact that genetically engineered microbes will rapidly spread in the environment and will contaminate organic farms. And so because all forms of genetic engineering are prohibited in organic production, the organic farming community is really going to need to grapple with this, and the onus should not be put on them for contamination. So just to look at how consolidation shapes innovation, this is looking at the realm of genetic engineering in crops. And that has been dominated entirely by a small set of major agrochemical companies. What you're seeing in this graph is the total acreage of genetically engineered crops grown in the United States, and 98% of that acreage is herbicide tolerant crops, Roundup ready, or engineered to be resistant to other toxic herbicides like dicamba and 2,4-D. So this is, you think of the promises that were made at the advent of genetically engineering crops about what could be done. But this is what we did with this technology, in part because of technological constraints, and in part because corporations will pursue applications of a technology that benefit them and not the public good or ecological good. So for the very abbreviated version of all of this, I would direct you to this Food Tank op-ed that touches on what's happening in this space, on these risks and concerns. And I'm going to leave you here, again, zooming way out, that we know that expert consensus concludes that we need to rapidly transition away from input-intensive agriculture to agroecological systems to address world hunger and the biodiversity and climate crises. So to really understand that pursuing the latest sort of shiny penny or silver bullet type of technology is not going to achieve the sorts of ends that we need, which is truly regenerating and protecting the soil, the water, and the climate, stable climate and biodiversity that food production depends on. Please feel free to reach out with any questions, and I look forward to answering some questions right now.
Thank you so very much, Kendra. Fascinating information. So we do have a few minutes for questions here. And so one came in just to back us up just a little bit on this term of biologicals. And when we're thinking about pest management, what is the universe of biologicals that play into this challenge of pest management?
So biologicals is an industry term to describe agricultural inputs that are derived from living organisms, juxtaposed with the synthetic pesticides and fertilizers that are currently dominant, that are derived from fossil fuels. And so I'm not sure I understand the question, because I think it's asking, maybe
Yeah. So
definition of the term, or
well, yeah. So when some of us not real familiar with the biologicals and what exactly that refers to, and if that is genetically engineered microbes that are used or could be used in agriculture, then that's the universe. We're just trying to understand
okay.
when we're thinking about pesticide use, what is biologicals?
Yes. So, zoom us back a decade ago, and there would have been no genetically engineered biologicals. And so when we used that term, we would be referring to naturally occurring microorganisms or pheromones used for mating disruption, anything, a compound derived from a plant. Most of these are approved for use in organic production. More recently, there is a set of genetically engineered inputs that for some, and from a regulatory perspective, they are falling under the definition of biologicals. And I think others would question whether they should be defined as biologicals or not, because they are not naturally derived, they're engineered. So I guess the definition right now is a bit complicated. because like the Office of Biopesticides under the EPA does include engineered microbes. It does include gene silencing RNAi sprays. And I guess my 10-year look back is not correct, because it also includes plants that incorporate a plant protectant. And so that would refer to genetically engineered crops, that BT crops. So that's a long answer to say, I guess it's a bit complicated. And it makes the regulatory sphere a bit confusing, because Friends of the Earth and other organizations would argue that the naturally occurring biologicals should have a different regulatory process than the engineered biologicals, but currently they are, falling under the same umbrella.
Okay, so you were talking about drift to farms that are trying to meet organic standard. Is it the engineered microbes that are the issue there?
Correct, because most right. I mean, a naturally occurring microbe on an organic farm, like this is this is the state of farming and organic farms, right? So
Yeah.
the question is genetically engineered microbes, because the organic standard prohibits all genetic engineering. And so this is a wide open question of how to deal with this problem, that as we have an increased amount of genetically engineered microbes in the agricultural landscape, that they are going to drift. And as I said, I think the most important thing is that the burden not be placed on organic farmers.
But at the same time, there's not much regulatory framework around those that do choose to use engineered microbes. Is there any,
Right.
controls to make sure that this drift doesn't happen?
There's not, we currently don't have any programs in place to track their spread or to evaluate impacts on the environment. Once they are commercialized and being used, they're just out there. And that's something we really need, is the investment in a system to really understand where are they going and what might potential impacts be. And currently, the different genetically engineered microbes fall under the EPA versus the USDA. So EPA governs any engineered microbe that acts as a pesticide, or that is transgenic, that is incorporating DNA from another organism. The trend has been towards using gene editing techniques like CRISPR, rather than creating transgenic microbes. So for anything else, if it's been gene edited and it's not a pesticide, that would fall to the EPA. And there aren't really regulations in place that take into account the unique properties of these engineered microbes. And so that's another place for growth, that we can develop regulations that are science-based, that are looking specifically at the properties, the traits, and the potential concerns related to engineering microbes.
Okay, well, it's a fascinating world. It is a place that we take for granted, this soil zone. There's a lot happening down there. And so this is really sounds like it has a lot of potential to maybe head us in directions of healthier ways to manage soils. But there is issues, and there's some growth that's needed here. So really appreciate you bringing this today as part of this discussion about how to find healthier ways to manage healthier soils. So thank you very much, Kendra. And I think we're about up on our time here. So
Right.
Appreciate
Thank
it
you. Yeah,
very
thank you so much
much.
for the opportunity.
Okay, and if you could also take a look at Q&A and chat and see if there are a couple more questions that you might contribute to. Thank you so much. All right, well, we are nearing the end of our time for today, but we want to remind you that this is just part one. We have a second session planned for Thursday, December 14. We're going to hear from Orion Valcourt on regenerating soils, from Catherine Daltrin, more on the microbiome as a mediator and target for pesticide-related human health effects. We'll get into the human biome a bit there. And then we've got an update planned for the end of the next session on what we're learning about the presence of PFAS in pesticide products. And this is not directly related to microbiomes, but we do have new information and thought it would be helpful for folks to hear an update in our next session. So that will be a wrap-up item for us, session 2. Really hope you'll join us again on December 14. It's going to be a great session.
Thank you so much, Greg. We're going to leave you today with a slide, one of MPenn's, well, several of MPenn's websites. And these are places where you can go to learn more about pesticides, and especially the pesticides and the Bay Watershed. Our flagship website is mdpestnet.org. It has hundreds of resources, including numerous research databases on pesticides, facts about alternatives, and a section on this program, the Pesticides in the Chesapeake Bay Watershed Project. And you'll find videos from previous conferences. And next week, we'll have the videos posted from today's presentations. Our Go Organic Now website has resources to help you vote with your dollar, vote with your fork. And that is by supporting organic growers, organic producers, and a healthy organic lifestyle. And the saferdisinfectants.org website is a project that was born right from this project's Healthy Alternatives Working Group. And it's where you can look up the safety rating for various disinfectants and choose safer alternatives. We also want you to please look for an email with a survey link, which we'll be sending out, and ask that you give us feedback on today's program. And thank you for joining us today. We hope we see you back on Thursday, the 14th for the final conference session. And lastly, I'd very much like to thank Greg Allen for doing a great job with our moderator, our terrific presenters today, and the team behind the scenes, Emily Ranson, Sean Lynch, and Doug Miller. Have a great day, everybody, and we'll see you next time.